So we don't lose time. I'm going to use a short introduction for Jenny Peterson, today's seminar speaker. Jenny is coming from the University of Delaware, where she just last September started as a assistant professor in the department, and get this, Department of Entomology and Wildlife Ecology. There's no ecology department in Delaware, so it's a combined program. Jenny had a complex career so far. So she has a BFA from she's an alumnus of Rutgers, BFA from Mason Gross, and she did her PhD at Princeton and done postdocs at Princeton and also at the University of Pennsylvania and Notre Dame, right? Alex Perkins. So she's interested also. She'd be happy to talk to any students that are interested in sort of non-conventional pathways to becoming an exceptional entomologist. And here we go. Jenny Peterson.
Great. Can everybody hear me? I don't know if this is picking up my voice.
Does it seem to be?
I just have a quiet voice.
It's right here.
Yeah.
Speak again.
Hello?
Yes.
Testing. Is that better?
Alex, can you hear? Jesse, can you hear?
Alex, can you hear? Yes.
Speak again.
Hello? Testing.
Is that better?
Kind of thing.
Okay, great. So thank you so much for inviting me and the introduction, Dina. So happy to be here. This is my first public talk since becoming faculty, and so this is exciting. So today I'm going to talk about some research I've done into multi-scale research into Triatomine Trypanosome systems, and I'll explain what that means. I have to figure out how to advance the slide here. Okay, yeah, so just a little bit, she already introduced me properly, but this is, in case anyone didn't hear it, this is my name and qualifications, and this is my hand, and this is my bug from Trinidad. So in case anyone forgot or didn't know, this is where Delaware is. I'll remind you of that. I didn't know until I did. But come on, let's be honest, it's a drive-through state, but actually we're doing pretty cool research there. So this photo really sort of conceptualizes why I do what I do, sort of like the conceptual underpinnings. And it's because I am so curious and fascinated by how something so small, like a virus or a pathogen, can sort of end up, in a situation of world domination. And I'm really curious about all the processes and systems that come together and interact to make that happen. And so the way I see it is, and I always emphasize this in everything I teach and every time I talk to students, is and I'm teaching conservation biology right now, and I talk about this a lot there too. It's all, for me, it's all about these systems and these interactions and the balance that is continuously thrown off, and then that effort to find balance, and it's this cross scales interactions between these pathogens and things we can't see, and then things as large as ecosystems, communities, etc. And so I really do emphasize cross scales because we're so limited by our own perspective. But really, in every system, there's all of these things happening and interacting. And so I'm really curious about different patterns, drivers, connections that all work together to form this phenomenon that we call disease. And working cross scales inevitably means cross discipline. So my title says medical entomologist, but it could easily say epidemiologist or ecologist, or because I just pull from whatever I need to. And so I was about to say jack of all trades, master of none. I should not say that. Master of all. Just kidding. So, but yeah, anyway, cross scales is cross discipline. And so today I'm going to talk to you a little bit about a couple of projects I've done in the past, and then I'll end with some work that I'm doing right now. So I'll start with investigations I did of pathogen-vector interactions, where I asked Does the vector get sick too? Are we the only ones getting sick? And then I'll move on to my work that I do in Trinidad, on the island of Trinidad in the southeastern Caribbean. And then I'll end with bringing it back here, work I've been doing on Triatomine sanguisuga in the mid-Atlantic. So starting with, at the smallest scale, do vectors get sick too? I don't need to tell all of you this, that what a diseased vector is, but I always include it because you never know when there's a member of the community in the audience or something. Diseased vectors, vectors that spread human pathogens, or if we're thinking about animals, animal pathogens. The way I came to ask this question, my training is in ecology. I got my PhD, my master's and my PhD in a department of ecology and evolution. And so really from that framework, we think of any infection as essentially an interspecies interaction. And so I asked, when a vector gets infected with a pathogen, it doesn't have to be a human pathogen, but that sort of brings it back to us because, humans were very human-centric. So that pathogen is inside the vector's body using resources. So does this come at a cost? I would think from an ecological perspective that it would. How could it not? Or, this is sort of the prevailing dogma, are vectors just sort of public transportation for pathogens, bringing them from host to host? So, and then, again, I probably don't have to go over this to this group, but, why study vector-pathogen interactions? Well, vector-borne disease, it's affecting a lot of people, and it's not just killing people, it's also taking productive years out of people's lives. And vector-borne disease incidence is increasing, and it's predicted to increase more with the triple threat of anthropogenic disturbance, global climate change, and biodiversity loss. And also, vectors are a feasible target. If we think of vector-borne transmission being sort of a product or having these three components, a very feasible target is the vector. It's a lot easier to go out and collect a bunch of vectors and knock them out than it is, a bunch of hosts. So, and then the case in point, of course, is this Wolbachia experiment, which I'm sure all of the people who study mosquitoes know. I mean, this kind of thing would never have been discovered if we weren't looking at vector-pathogen interactions. And so this is the famous Wolbachia that you inject the mosquitoes, and then they, the Wolbachia outcompetes the dengue, and the mosquitoes aren't spreading the dengue anymore. So I asked then, does Trypanosome cruzi affect kissing bug fitness? And I'm going to go over this system that I use to test this question of are the vectors affected by infection? And so the experimental system I have used thus far is the Chagas disease system. And Chagas disease is spread by triatomine bug vectors, which are Hemiptera, and known as kissing bugs. And the parasite is Trypanosome cruzi. And so the disease itself, it's kind of a slow, silent disease. It makes you really sick right at first, and then the parasite kind of, your immune system can keep it down to a certain level, and then it gets inside your cells and it slowly starts reproducing. And then every time enough of them inside a cell reproduce, it explodes the cell, and then, and so it causes this repeated inflammation over time. So the disease eventually, over many, many years, can lead to serious, I should say, unfortunately, the parasite's favorite tissues are cardiac tissues, gastrointestinal tissues, sort of these important tissues in our body. And so over time, it can lead to these serious cardiac or gastrointestinal alterations, sometimes resulting in death. And really, since Chagas is a slow disease and it's often a silent disease, it's not usually, its impact is best measured in disability-affected life years. So if you look at all of the parasitic and vector-borne disease statistics from Latin America, you can see that Chagas disease, it takes the most amount of productive life years out of people's lives. So often they say people don't die of Chagas, they sort of live of Chagas. It works better in Spanish. But but that is what they say, and so that's really a big argument for studying this disease. So I asked, does T. cruzi make its kissing bug vector sick? And to do this, I used a really basic experimental design. I took a bunch of, I did all this in Medellin, Colombia, and I took a bunch of Rhamnus prolixus vectors and I infected them with Trypanosome cruzi. So this is, also I should say, this is kind of the basic design, and then there's some nuances I'll get into in a second. So, and then I monitored their survival, the vector survival, for 90 days, and then I did a survival analysis at the end. So, Trypanosome cruzi, if anybody knows anything about it, it's a very, very diverse parasite. And it's because it has this very large geographic range, and then it has all of this environmental variation that's then driving variation in the parasite. So it infects over 150 vector species, it infects hundreds of mammal hosts, and it has an expansive distribution. And it also co-infects with other parasites. And so these are all sort of drivers of its diversity. And so I wanted to capture some of this genetic variation of T. cruzi in my experiments. And so I didn't just infect the vector with T. cruzi, I infected it with 5 different strains of T. cruzi. And I wanted to see if there were variations in the effect that it had on vector survival. And so I infected them with 5 different strains, and then I recorded survival, or we can say death, same coin, for about 4 months. And then I compared survival of the infected groups to a group that was not infected. And so this is what I found. And the biggest take home with this graph here, we'll have, this is infection day, day 0, and this is how long they lived. And each of these represents a treatment group that was infected with a different strain, and then this is the proportion alive at any given time. So really the take-home message here, the things that I'm interested here, is kind of the big differences here. So essentially what I saw was that after, towards the end of the experiment, people, I call everything people, sorry, the bugs were infected with, that were infected with this strain 5. They were doing, more of them were alive than in the control group. Whereas the bugs infected with strain 1, they were almost, they were at about 20%. And then the others were just sort of in the middle. But basically the take-home message of this is that yes, there is a cost. There can be a cost, but not always. And so I wanted to test this again and see, kind of get more information. And so the next thing I looked at in this experiment, in a subsequent year, was I wanted to understand if co-infection had any effect on this survival. And so this time I infected bugs with 2 parasites, Trypanosome cruzi, and then I also infected it with this parasite, Trypanosome ranhelii. And Trypanosome ranhelii, it's a sister species of T. cruzi, but it's important because it co-circulates. So it infects a lot of the same hosts. It infects most of the bugs of the genus Rhamnus. And so yeah, it's a sister species, co-infects. Yes, so it has this inverse pathogenicity situation though, which is where it's not pathogenic to humans. And the way this was discovered was back in the, I don't know, we could call it the golden age. Some people were like, I don't think it's pathogenic. Yeah. And that's how they discovered it. Oh man, love those old papers. So that's how we know. And but it is, or it was believed pathogenic, to be pathogenic to the vectors. I have a paper where I sort of refute this, but we don't have to go there right now. So in the experiment number 2, I did the sort of same idea. I had different treatment groups. I had bugs not infected with anything, just fed blood, and then I had bugs infected with just T. cruzi, just T. ranhelii. Whoops, sorry about that. Just shouted into the microphone. And T. cruzi, T. ranhelii co-infection. And I monitored vector death. And then this time I also wanted to do fitness calculations, and so I monitored reproduction. And so here I found, this is just showing the treatments of insects. The black is the control, so that's what we're comparing everything to. I should have said that in the last one, sorry. So here we are just looking at the relationship between the black line and then the other two. And interestingly, it seems like both, first of all, it doesn't seem like they're that different, which is surprising considering that if you remember, T. Reinhelli is supposed to be pathogenic to the bugs, and while T. cruzi is supposedly not. And yet both of them had a very similar effect on survival. But the interesting thing came when I looked at the co-infecteds, and for some reason they seem to do better. And I don't know why this is, and I even tested it. I'm not going to show these data because it's one of those like never-ending things that keeps getting pushed aside and then brought back up, so it's still there. But I have other experiments where I saw the same effect. I tested dose, I tested infection timing, in terms of like if the co-infecteds, if they were infected with T. cruzi first and then T. Reinhelli, or T. Reinhelli first and then T. cruzi, but I almost always saw this result. And so I don't know what's going on there, but I think I also saw the same in reproduction. The co-infected insects, this is their reproductive score. It's calculated based on essentially the number of eggs they lay is sort of a function of how much blood they had eaten at their last blood meal. So the mixed, there didn't seem to be a negative effect on the insects that were co-infected, and then the fitness estimates also showed this again. And so back to my research question, do disease vectors get sick? You know, and here I just kind of, that's the hook. Really, it's, you know, the specific, the life history and the fitness and the survival and all of that. But it seems like essentially, yes, but it's complicated, as always. It's highly variable. It depends on parasite co-infection. It depends on strain. So I do think that we can't definitively say, yes, it's this, it's always going to be this. But that's sort of life for you. But I don't think they're just pathogen transport. And I am curious if the negative effects of the mono infection were sort of somehow ameliorated by the co-infection. So if I were teach a lot. If I were teaching, I'd ask for questions now, but I think we'll do that at the end. So I'll just move on now to investigations that I've done on Trinidad. So shifting gears here, this is some of my work that I've done on an island that is not considered to be endemic for Chagas disease. So this is from the World Health Organization, and all of these red dots here show how many people are infected with T. cruzi. And all of these white dots here are no data. No data for any Caribbean island, even though they have triatomines. Here's the Caribbean up close, and here's all the places with triatomines. Yeah, there's no data. And so, and the official message, if you look in all the publications and everything, is there is no Chagas transmission in the Caribbean islands. And so they say T. cruzi transmission is strictly enzootic. But the flip side, this is sort of my response, is, well, you haven't really looked. And then few clinical diagnoses. And again, you haven't really looked. So I wanted to really look. So I went to Trinidad and Tobago. And this, in case for anyone who's needs a little refresher on their Caribbean geography, Trinidad and Tobago is a dual island nation, and Trinidad is pretty large relative to the other islands around it. It's got about 1 million people, and as you can see, like, they're one nation, but Trinidad's much larger, and most of the population lives on Trinidad. It's got an interesting ecological history because it's kind of just a piece that broke off the South American continental shelf, and this part up here is only it's less than 7 miles from Venezuela. So it shares a lot of the same flora and fauna with Venezuela, including 6 triatomine bugs species. So, and Chagas disease has sort of been studied on and off on the island. I'm not the first person to go, wait a second, In the 60s, there was a cardiologist who suspected that some of his patients had Chagas disease. And he tested them for T. cruzi, and he found positive serology. And so he went to their homes, and he found hundreds of vectors in their homes. He found 50% of them to be positive for T. cruzi. He sort of completed the cycle for everybody, and nothing happened. And then as someone picked it back up in the 80s. They went and they described a couple new species for the island, but again, nothing really happened. And then in the 90s, again, someone picked it up, found more cardiac patients infected, and then nothing happened until we picked it back up in 2016. And so in 2016, we essentially felt like we had to kind of start from square one in terms of like, is this really happening, or was there a reason? Was there something wrong with those studies that nothing, it's still like no data by the World Health Organization. So we just started with some very basic research questions because we had a very small budget, and we asked, are the vectors still there? Where are they? Are they near people, which is usually, what we're really asking when we say, where are they? What's the T. cruzi infection prevalence, and who are they making contact with? So essentially, who are they feeding on? And so we had a simple experimental design. We sampled the bugs throughout the island in a pretty short amount of time. We mapped the positive collection sites and then processed them through microscopy. And then we did blood meal analysis. So this is where we sampled on the island, and we kind of divided it into these different landscape types. So we looked at these residential landscape types, or sort of peri-urban, residential rural, so like a home that was sort of surrounded by maybe like an abandoned cacao farm or something like that, and in forest patches that were sort of near built environments. And Trinidad has a lot of sort of nicely forested areas, but they all are still patches, and deforestation does continue to happen there. So for the vector sampling, we tried out, a lot of times with triatomines, you kind of just gotta like throw everything at the wall and see what sticks, because you don't know what's gonna attract them. So we did mouse-baited traps, we did manual collection, we did community collection. And the mouse-baited traps, I don't know if anyone here has ever done mouse-baited traps. It's hell. But I found out that I'm not able to tell the gender of two mice because I made baby mice. I didn't mean to. I thought I was separating them. Yeah. So anyway, it's a funny little system. So you just go to Home Depot and you get some PVC pipe and you wrap it with double-sided carpet tape. It's very sticky, and then inside you put like a little, mouse furniture and food and all that stuff, and then you put the mouse in. And you set it somewhere where you think the bug might be, and then the bug lands on it, like that bug, and it gets stuck. So we tried that. We caught a few that way. I won't, hold on. I'll get to how many they caught in a sec. But yeah, so that was one of our methods. Manual collection, it's a fancy way to say we just looked everywhere. And then community collection. So, and the community collection wasn't planned ahead. It was something that we discovered when we were interacting with people as we were setting traps or talking to people, and we found out there was just a lot of curiosity, and there really wasn't any resources for them in terms of the bugs there. So we repurposed our urine cups for community collection, and we gave them the cups and gave them a glove and instructions. And we also gave them this flyer. Oh yeah, I was like, is that a note to myself? No, I actually did give a seminar. I gave a seminar on how to differentiate vectors from lookalikes, because as you all probably know, there's a lot of bug blindness. And so the bug is about this big, and I would get calls for like, there was a bug that was like this big. I'm like, no. So, and also people will find like a milkweed bug or an assassin bug that's not a kissing bug, and they'll panic. And that's really what we want to prevent, is the panic. And so that's why I did that seminar. And so then to diagnose T. cruzi in the bugs, these bugs are really special in the way that they have this stercoral transmission. So essentially it comes out in their excrement, the parasite. It does not come out in their saliva. And so to figure out which ones are infected, you just take a little bit of their poop out and dilute it with PBS and look at it under the microscope, and you can see the trypanosomes dancing. It's really cool, but also that doesn't always work. Sometimes if there's a really low level of infection, so you can also confirm it with PCR. And then we also sequenced a couple T. cruzi amplicons to make sure that it was the Trypanosoma cruzi, because there's a lot of trypanosome species. Yeah, and then to figure out who they're feeding on, we did blood meal analysis, and probably anyone who works on vectors knows what it is, but anyone else, essentially we extract DNA from the bugs. Ideally it would be their guts, and then we run it in a PCR with a primer that targets a universally conserved vertebrate sequence, or gene sequence. And so then we sequence the PCR amplicons, and we blast those in GenBank, and then we can essentially figure out which hosts the bugs we're feeding on. So vector blood meal analysis, it gives a lot of information, not just on who they're feeding on, but then it can tell also which landscapes these bugs are in. And that's important for us because we want to know if the bugs are near people. And so if the bugs are feeding on not only people, but if they're feeding on dogs, cats, chickens, things that tend to be around people, we can infer, okay, they're in areas where people are. And that's epidemiologically interesting. Whereas on the other hand, if they're feeding, only on howler monkeys, we can assume that, there's no people up there with them. So, and then also the host types, as I was saying, that's kind of just what I said. It's interesting to see the host types. If you want to think of, these two are the ones associated with humans, and the wild animals would presumably imply more of an enzootic cycle. So it was a pretty short expanse of time, but we found 55 triatomine bugs, and 54 of them were this species here. These are all our pictures from the trapping, and this species is Panstrongylus geniculatus, and then we found one Rodney's PT phase, and we found only adults. That implies that they're foraging, because only the adults can fly. So yes, there's still vectors in Trinidad, but where were they? So all of our positive collection sites were in human residences. They were in, inside the house and outside the house. Let's see, do I say this? No, I don't. Okay. So none of the sort of forested sites, we couldn't find them there. That doesn't mean they're not there. It just means we didn't find them there. We did find them though in only in human sites with humans, used by humans. So, and then essentially, most of our positive sites were in the north and central part of the island, but that's just sampling bias there since we only had one in the south. Oh, here we go, the landscape types. Okay, so yeah, we had the residential, peri-urban, 2 of 3 sites positive. Most of our sites were residential rural, so like a house and then a forest patch and another house. And 4 of those were positive for bugs, but none of our forest patches were. And so then we found about an 84% T. cruzi prevalence in the bugs. We found bugs infected with T. cruzi at all the collection sites. And then this is a Giemsa stained Trypanosoma cruzi from Panstrongylus geniculatus poop. And it's just, they're just such beautiful parasites. I love that picture. And it just luckily had situated itself right by the scale. I was like, thank you. So these bugs, when they're infected, simply from a sort of a science, anyone who loves science, it's just so cool. There's so many parasites. And when you look at this under the microscope and you think that they're all moving, it looks like your slide is shaking. It's just really a cool experience. I think everyone should get to do it. Okay, so what hosts are they feeding on? So we found through our blood meal analysis, we found that they were feeding on 8 mammal species and 4 bird species. So we, this was low budget, so we only had money to do Sanger sequencing, which essentially we could identify 1 blood meal per bug. Blood meal analysis has gotten much, or is, it probably was sophisticated then too, we just couldn't do the expensive thing. But Illumina sequencing now, you can get like multiple blood meal hosts per bug. But basically, as you can see here, it looks like they're munching on people quite a bit. The humans were by far the most common mammal species, which isn't incredibly surprising considering that we found them, in people's homes. But the other interesting thing is the rest of the mammals are wild mammals. And so, but you got to remember these people are living their houses are abutted against the forest. So to me, this paints a picture of, the bugs are probably breeding in the forest patches, and then when they can't find food in the forest, they go to the humans, and they go to them pretty regularly. And the bird species kind of had the same pattern. There's not wild chickens there, and so chickens would imply also that they're hanging around people, but then also we have these wild birds. And so, and just in case anybody doesn't know, Trypanosome cruzi, it's a mammalian parasite, and so birds serve as they serve to feed the vector population, but they're dead end hosts for the parasite, and so that's why I separate them between birds and mammals. So here's just a breakdown of the host summary. So we found that it seems like they are feeding on humans and chickens a lot relative to the other wild animals. So current status of Chagas in Trinidad after this. We thought, okay, infected vectors are making contact with people, definitely. People are likely being exposed to T. cruzi, but we don't know the frequency, and so we need more needs to be done. And so at this point, and I'll just say Panstrongylus geniculatus, this species is important because it has this incredibly wide distribution, and it's proven itself to be a very adaptive species. So in the past, it was just kind of strictly associated with armadillo burrows. But recently, like in the past couple of decades, it's been found all over Latin America in metropolitan areas. And it's been found to breed in homes, invade human homes. It's just very adaptive. And so that in itself is sort of an epidemiologically interesting characteristic. The other thing is T. cruzi can be transmitted orally. So these bugs are hanging around the kitchen, and you're making juice and one falls in. Oral, that actually has happened a few times, and it's been served to children at schools, and it's just, oral Chagas is a really serious transmission mode because the parasite dose that you get on oral Chagas is orders of magnitude larger than you'd get from just a little bit of poop on the bug that you then scratched into your skin, and it's much more serious. And so, and Panstrongylus geniculatus is very often the species for some reason. I don't know if it likes fruit too or what, but it is a problem not just for vector-borne Chagas, but for oral Chagas as well. So, and then after seeing this, I poked around a little bit more, and I looked at the blood bank prevalence. So this is, a Pan American Health Organization. The countries of Latin America and the Caribbean, they give their blood bank data, or they take the blood bank data from all the countries in Latin America, and so one of the things they test for is T. cruzi prevalence in blood donors. And these countries all here are in Central and South America, and they're all considered to be Chagas endemic by the World Health Organization. And this was their blood bank prevalence of Trypanosome cruzi infection. And this is Trinidad. And so it just made me feel more and more like, okay, we need to research this more. We need to find the evidence to make the case that World Health Organization and other global health organizations need to take this situation seriously. And why is that? Who cares about status? We don't care about status. We care about money, right? And that's really what it amounts to. Is once you're endemic, you get drugs, you get diagnostic tools. Two things that don't happen if you're not endemic. And so that started the project that is ongoing now. My collaborator after this decided, he's a veterinarian at the University of the West Indies. He decided he wanted to get a PhD and he wanted to study this. And so now he's down there working on this, and I'm his advisor, and this is the project that we're doing. And so this is, some of this stuff has already been done, but the data is still being analyzed. And then some of our latest findings are, since he's a veterinarian, the animal stuff was the stuff that came in first, but I always think these are kind of fun. So this is all these these are a mastigotes and an armadillo heart, and the armadillo has myocarditis, so it has like heart failure. Here's T. cruzi and canine tissue, and then here's T. cruzi and canine blood. So we're finding it in the animals too. So that's where that is now. And so now I'm going to shift gears to end with a talk about some of the more recent stuff I've been doing since I started at University of Delaware in September. So looking at our local species, Triatomine sanguisuga, in the Mid-Atlantic. Chagas disease, and I didn't say this in the beginning, I should have, is generally, it's vector-borne only in the Americas, but it's generally considered to be a Latin American problem, and it's sort of associated with, emerging market countries. But we have them here. And these are the 11 species that we have here in the United States. If you count, you'll see 12, because for some reason this photo includes 2 subspecies. And these are all the states where the bugs are distributed, or where they've been found. And so they're pretty ubiquitous. I mean, across, and this really, I'm really curious about here and here. I think there's probably bugs there too. But what I want to talk,
Sorry.
whoa, microphones and me. Okay, so I'm going to talk about T. sanguisuga right there. That beauty. It's a really beautiful species. So T. sanguisuga is distributed. It has the widest distribution of any triatomine species in the U.S. It's distributed essentially from the Rocky Mountains all the way to the East Coast. And studies of T. sanguisuga, though, have happened in those states. So there's at least 24 states, and we've studied them in 3 of them. And yes, we can get good information from 3 states, but I don't think necessarily what's happening in Texas is representative of what the species is like and its different different complexities and ecological characteristics in the coastal forests of Delaware. And so this really surprised me, but not really. And what we know about sanguisuga from the few studies that have been done in the Gulf states, we know it's a generalist feeder. We know it's highly adaptive, and we know it transmits T. cruzi. And I'll just give a couple examples because I think these are cool. So these are in New Orleans, rural New Orleans. Although I think I read a study where they were found like near Bourbon Street. This is a one blood meal host study from 2014. And again, take home message here, they feed on a lot of different species, including humans, but also frogs. They're just so adaptive. It's so interesting. And this one, I know it's a lot, and I almost deleted this slide, but I really like what it's showing because this shows the different feeding networks that they took bugs found in a shelter, and they created interaction networks based on their blood meal analysis. And I mean, look, it feeds on these lizards. It'll essentially, if you have a heartbeat, it will eat you. And the house bugs too. These are bugs. This is just bugs found in human homes. And I mean, they'll go and eat anything. And then finally, this this is from bugs from Texas. And these are not the species I'm studying, just these. But again, these people did the multiple blood meals in each bug. And I mean, they'll feed on, like, that was from a zoo. They were in a zoo feeding on tigers. They don't care. Take them to a restaurant, they'll just take anything on the menu. So really, that just got me asking, well, what's going on here, But maybe we don't care. I don't know. Maybe it's just completely epidemiologically irrelevant. Then 2019 happened. And at that time, the CDC keeps, I was really hoping to find like the CDC and triatomine maps from like 2015 to now, because every year they add a new state. And in 2019, Delaware was not on it. But then my friend Triatomine flew in a little girl's bedroom, bit her on the face while she was watching TV, and the media went wild. By the way, Delaware's here. This was my Google. I just Googled this the other day, and it's just like they just caught it, they spun it, they posted photos of the wrong bug. It was insane. Really. The bug's been here the whole time. I just went and searched the different citizen science sites, and I searched some collections, and I searched some old emails of Charles Bartlett, and I put together all the places that it's been found in Delaware. And I feel like I got there. I got to Delaware in September, and someone just said in passing to me in the hall, "Oh, they found a kissing bug in someone's house in Smyrna," and I think I wasn't supposed to say the city. Sorry. Anyway, yeah. So they did, but I followed up on that, and I was like, "Well, where is it? What happened?" And they're like, "Oh, it wasn't a kissing bug. It's fine." And I was like, "Can I see it?" And I was like, "Yeah, that's a kissing bug. Did you tell the person who lives there?" And they're like, "Yeah, we told her it wasn't a kissing bug." I'm like, "Oh dear." Okay, so called her up. The person happened to be a medical professional who had been incredibly diligent in trying to find out what this bug was. This person called the CDC, called pest company, called their primary care physician, called infectious disease physician, got zero answers. And so I call her, and she told me that, and then she said, "Oh yeah, and we found another one on my favorite cookie sheet last night." So I went, I took the bugs from her, I tested them for T. cruci, and ended up finding the first T. cruci in Delaware. So, oh, by the way, I should have said it was under her pillow, because it was. And also then I worked with her to find, because when she had found the bug, she was just getting over COVID. Oh, they were just getting over COVID. Okay, just fire me now. And they're having heart palpitations. And so, and then finding this kissing bug, and this is a medical professional who knows that kissing bugs are related to cardiac, even though the timing would have been totally wrong. But anyway, this person was very, very, very worried. And so after I found out it was infected with T. cruci, I worked with them to finally get T. cruci serology done. And it was negative, so that was good. I mean, it's not surprising that it was negative. You don't generally, unless you're the most unlucky person in the world, get T. cruci from one bite. So is Chagas coming for us? Well, autochthonous Chagas in the U.S., so vector-borne Chagas, 1955 to 1998, 5 cases. Then the next 20 years, 78 cases. 15-fold increase. What's going on? So, Chagas is surveilled in the U.S. in 6 states only. So, Massachusetts, you'll count 7 because Massachusetts bowed out in 2014. So 6 states only, okay? 74 of these cases came from the states with surveillance. And this is the thing with Chagas. This, I think, is why so many of us find it so intriguing. If you look for it, you find it. If you don't, you won't. Generally. We weren't looking for it, we started looking for it. The internet was born, So, people started being able to look stuff up easier. So do I think it's coming for us? No. Do I think we need more information? Yes. So this leads me into what I'm doing now, which is I'm just trying to get this baseline ecological and epidemiological information about Triatoma sanguisuga in the Mid-Atlantic. And I'm starting with lovely Delaware. Do we remember where Delaware is? So this is I have a little pilot grant that is going to be helping us, me and my grad student. We're going to go all over Delaware this summer, and we're going to sample it. We're going to figure out who it's feeding on, T. cruzi prevalence, just essentially figuring out where it is. But we want to know, as always, in relation to humans. Is it, Delaware is an interesting place, I'm discovering, because it's a matrix. It's really, I feel like it's like where most residential areas were like 50 years ago in terms of its level of development. Because where I'm from, it's all paved over, but Delaware is still in that process where it's this matrix of like farm, forest, neighborhood, farm, forest, neighborhood. And so it's kind of is the perfect setting for these bugs to go back and forth. And especially these bugs, as I showed in the blood meal analysis, if they're anything like the ones in Louisiana, they love frogs. And Delaware is like mostly flat, it's mostly a swamp. There's tons of frogs. And so, it's got sort of abundant food sources. And so it just kind of seems like a great place to be Triatoma sanguisuga. So this is what we're doing. And I guess it's, and our objectives are to collect this baseline information. And really it's so that this poor person who could not get information simply on the species of this bug, that shouldn't happen with the internet and all the resources available. And it really, to me, it was no particular person's fault or entity's fault. It just, to me, clearly illustrated a gap in the system. And that's a gap that, luckily, I'm getting paid to fill those gaps. So that's what we're hoping to do. To be continued. And then these are all the people that have kind of helped me along the way. Yay, we have time for questions.